Assalamualaikum and good morning to the panelists and my fellow members. I'm Syazana Adilah Binti Zulhaidi, a final year student in Bachelor of Science Honours of Applied Microbiology. The topic for my research proposal is BIOFILM inhibitory agents targeting predominant oral pathogens. Throughout this presentation, we will explore 7 key subtopics. Let's go to the study background. The oral cavity is a dynamic and diverse microbial ecosystem harboring a multitude of distinct microenvironments and microbial communities, making it the second largest in the human body, comprising over 700 species. The oral cavity serves as a thriving environment for microbial growth, particularly on two main surfaces, the hard tissues of teeth and the soft tissue of oral mucosa. When the balance of oral microbiome is disrupted, these biofilms can escalate into pathogenic states, leading to oral diseases. Biofilms, as described by Simon et al. 2019, are intricate microbial communities embedded within an extracellular polymeric matrix composed of proteins, polysaccharides, and DNA. Microbes within biofilms exhibit heightened resistance to antibiofilm agents compared to planktonic cells. Therefore, the identification of natural agent with biofilm destructive properties holds promise for combating oral infection. For instance, essential oils from Lipia alba and Lactobacillus plantarum lipoic acid effectively eradicating microbial biofilms formed by Streptococcus mutans. Dental biofilms are a major concern linked to issues like periodontal disorders, candidiasis, and dental caries. To effectively address these oral diseases, we need anti-biofilm substances capable of selectively targeting and dismantling oral biofilms. Dentistry relies heavily on antibiotics like penicillin, cephalosporins, and metronidazole, which has led to drought resistance and disruptions in oral microecology. The overuse of antibiotics has created selective pressure on the oral microbiome, eliminating susceptible bacteria and fostering the growth of pathogens armed with antibiotic-resistant genes. Hence, there is a growing need to identify novel antibiofilm agents specifically from natural sources. The objective for this study are to identify the predominant oral pathogens that form biofilms in the human oral cavity and their roles in oral diseases, to determine the plant and microorganism-derived biofilm inhibitory agents and their effectiveness in combating oral pathogen biofilm, and to review the mode of action of plant and microorganism-derived biofilm inhibitory agent in destroying the biofilm formation. This review holds significant importance as it addresses the urgent need to combat biofilm formation by prevalent oral pathogens. Assessing the effectiveness of this biofilm inhibitory agent is key to advancing oral health management. This review shines a bright light for researchers to develop more effective strategies preventing and treating biofilm-related oral conditions, ultimately enhancing overall oral health and well-being. Now we move to literature review. Oral cavity. A species-rich heterogeneous ecological system comprises distinct small microbial habitats including the tongue, teeth, buccal mucosa, soft and hard palate. A moist, warm and nutrient-rich environment is optimal for microbial proliferation. When the oral microenvironment or oral hygiene practices undergo alteration, opportunistic microbes may proliferate within the oral microbiota. As I mentioned earlier, oral biofilms are groups of microbial communities that are organized structurally and physiologically. Microbes within biofilms are shielded from UV light exposure, dehydration, host immune cells, and lethal molecules by a complex microenvironment. As you can see from the figure given, biofilm formation can be divided into 5 stages. Saliva transport crucial components which important in maintaining homeostasis inside the mouth cavity as well as excellent oral hygiene. Decreased salivary flow rates and poor oral hygiene will promote the growth and build-up of bacteria that are more resistant to acid, which contribute to oral infection. Oral pathogens are microbiota that reside within the oral cavity and are implicated in a multitude of conditions and diseases that impact the tissues of the mouth. Streptococcus mutans, an oral Streptococcus species classified as gram-positive, facultative, and aerobic, has been identified as a significant causative agent of dental caries. Excessive sucrose consumption by hosts induce a sequence of structural and ecological changes that promote the development of S mutants. S mutants secreted mutations which will eliminate adjacent competitors. Next, Porphyromonas gingivalis, a black pigmented gram-negative anaerobic bacteria, is a major etiological agent in periodontitis. It has a wide range of various factors such as fimbriae, capsules, lipopolysaccharide, and more. In addition to bacteria, Candida albicans is the most prevalent member of human microbiota, followed by Candida tropicalis and Candida glabrata, which are associated with candidiasis, early childhood caries, and even mucosal malignancy. Candida albicans can be found in the human microbiome as yeast. The change from yeast to hypha is a pathogenic transition. Lastly, anti-biofilm strategies. It is estimated that biofilms cause 65% of infections, which are challenging to cure. Therefore, finding potent anti-biofilm agents with low cytotoxicity is essential. The optimal anti-biofilm strategy should not only eradicate pathogens and inhibit new biofilm formation, but also facilitate the dispersion of formed biofilms without disrupting commensals that could lead to microecology dysbiosis. The optimal approach for inhibiting biofilm formation appears to be focusing on either or both of the first two stages of biofilm formation. There are various types of agents that could control oral biofilms, including quaternary ammonium salts, small compounds, arginine, and natural products. Numerous natural compounds derived from plants, microbes, and marine life have demonstrated anti-biofilm properties. For example, curcumin and tea extracts have excellent anti-adhesion properties against several gram-positive oral bacteria such as Streptococcus species and Staphylococcus species.
Next, the Gantt chart here shows the timeline of the various steps of this project. It helps me to plan and track the progress of the project. Overall, I started working on my proposal on October and it will be submitted on this January.
This is the cited references supporting my statement. So that's all for my final year project presentation. Thank you.